Hello everyone, nice to meet you all. I'm Zhou from China, Shandong University. Today it's a great honor for me to participate in this meeting. Thank you very much, my dear friend Dr. Rao, for inviting me to give a speech here. Today, I'd like to share with you about one of my papers. The title is Sequential Activation of Uterine Epithelia IGF-1R STAT3 Signaling by Stromal IGF-1 and embryonic IGF-2 Conquer the epithelial barrier for successful implantation. First, let me introduce you some background knowledge. The pregnancy timeline in human. When women produce a mature oocyte, the oocyte will get into the fallopian tube. On the other side, if the sperm cells show at right time, they will meet in the fallopian tube and fertilization occur. Then the new fertilized egg will develop through 2-cell, 4-cell, and the molar stage to become an early blastocyst. At this stage, it will be transferred from the fallopian tube into the uterine lumen, where only the blastocyst build an interaction with the uterine wall. It can continue to grow, otherwise the blastocyst will be lost. This process, we call it the embryo implantation. After implantation, it's the initiation and formation of functional placenta, and the continued placental and fetal growth until parturition after 35 to 40 weeks. Our research field is about how the implantation happens. After many years study on it, now we know there are 3 prerequisites for successful implantation. Firstly, it requires a functional embryo, which means the embryo should be at the blastocyst stage, and second, a receptive endometrium, and then the synchrony between the embryo and the uterus. Here, you may ask, what is a receptive endometrium? As we know, the embryo is half allogenic for its mother. So during a menstrual cycle, uterus only allowed an embryo to implant in a very limited phase. We call this time the implantation window. And at this time, the endometrium was at the receptive stage. For human, it's around day 7 after ovulation, and in mice, it's on day 4. Then we can raise a question. How does the uterus know when to enter the receptive stage, and how to make sure both the uterus and the embryo develop coordinately? Previous studies prove that there are 2 important hormones playing roles during this process. They are ovary-produced progesterone and estrogen. For example, on day 4 of mice, the progesterone dominate the uterus until a small wave of estrogen comes, and estrogen work opposite to the progesterone, stop the uterine epithelial cell proliferation, and transit it into differentiation to build the receptive stage. And how does the estrogen promote uterine epithelial differentiation? We now know that estrogen could activate its receptor, alpha, in both the epithelium and the stroma, and the stroma cell would produce some Pgr-Cre factor to stimulate the epithelium. Here, IGF-1 is such a Pgr-Cre factor. IGF-1, its full name is insulin-like growth factor 1. It activates its receptor IGF-1R to promote uterine epithelial cell proliferation, but this happens in the non-pregnancy mice. Then we wonder whether IGF-1 is also important for implantation. And IGF-1 is a ligand of the insulin-IGF system, and besides IGF-1, there is another ligand, IGF-2. Both IGF-1 and 2 work through their common receptor, IGF-1R. Here, to explore the function of IGF system during early pregnancy, we first check their expression. These 2 ligands in their MR level. we could tell that IGF-1 was expressed in the stromal cells of uterus, but not in the epithelium. On contrast, IGF-2 was not located in any uterine cells before implantation, but during implantation, it was highly expressed in the embryo. Further, the study also showed that receptor IGF-1R was mainly located in the uterine epithelium, indicating that IGF system may play a vital role during the implantation process. Next, to explore the function of IGF, we adopted a Pgr-Cre mice model, which could efficiently knock out the target gene in uterus. So we meet the Pgr-Cre mice with IGF-1R fluxed mice and notice that IGF-1R was absent in the uterine epithelium, and the knockout of IGF-1R was very efficient in both the mRNA and the protein level. Then we check the results of IGF-1R deletion. And the knockout IGF-1R lead to a decreased implantation risk. You may see the blue spot was less in the IGF-1R KO mice we marked as DD, compared with the wild-type ones we marked as FF. And the implantation markers like COX-2 was also reduced in the IGF-1R KO mice, which represents that IGF-1 signaling is vital for implantation process. Next, we want to know why the IGF-1R deletion impaired implantation. Does it influence the progesterone and estrogen level? Because this mice model is also work in the corpus luteum of ovary. But the examination results of female serum prove that IGF-1R deficiency does not influence hormone synthesis. The progesterone and estrogen level of serum were comparable between those two groups. But it did affect the estrogen responsive in day 4 evening of the KO mice. And as a result of the dysregulation of estrogen responsive, we could tell the tight junction members expressed highly in the epithelium of KO mice, which blocked its transformation and differentiation for successful implantation. Here, we can summarize it as uterine IGF-1R mediated signaling influence pregnancy rate by regulating epithelial transformation during peri-implantation. And because IGF-1R is a receptor, then we want to know what's the downstream of IGF-1R in the uterus. There are 3 classical pathways of IGF-1R, and we noticed that JK-STAT3 pathway was activated during peri-implantation, and the activated STAT3 was gradually increased in the epithelium on day 4, and it is still sustained in the epithelium around the implanted embryo on day 5. And the IGF-1R deficiency decreased the activation of STAT3. So we raised a hypothesis that combine the expression of the ligands with hypothesis at stromal-derived IGF-1 ligand may regulate the whole epithelial transformation via STAT3, while local epithelium around embryo was under control of fetal IGF-2 ligand through STAT3 pathway. To prove this hypothesis, we designed a 2-part experiment. In the first part, we remove the ovary and give the mice estrogen to induce IGF-1 expression. And we notice with the increased expression of IGF-1 intrauterus, you may see the activation of STAT3 was also increased in the epithelium. But compared with the wild-type one, we could tell IGF-1R deleted mice showed the absence of STAT3 activation, and the epithelium differentiation was also impaired. So we proved that the first part of our hypothesis, that stromal IGF-1 activated epithelial IGF-1R to mediate signaling function, mainly through STAT3 pathway in the epithelium. Next, we checked the expression of IGF-2 in both the unimplanted and implanted embryo, and found that IGF-2 was rapidly increased during the implantation process. So we flushed the unimplanted blastocyst from uterus and block it in vitro with IGF-2 specific antibody, and then send them back into the uterus to check whether they can implant or not. The statistical results show that blocking IGF-2 affects implantation rates, which means specific blocking fetal IGF-2 could prevent embryo implantation. On the other side, we coated the IGF-2 recombinant protein on the surface of beads to mimic the blastocyst, and transferred the beads into the uterus, and found IGF-2 coated beads could activate STAT3 expression, could activate STAT3 activation and induce the implantation marker COX-2 expression. So here we summarize it as fetal-derived IGF-2 locally activated IGF-1R to promote implantation via STAT3 pathway in the epithelium around embryo. And as some people reported that as a transcription factor, STAT3 could bind on the promoter of COX-2 to induce COX-2 expression. So we want to know whether it's the same in the uterus epithelium. So we did the chip experiment with STAT3 antibody and proved that in the epithelium, STAT3 could bind in the promoter, the primer 1 region of COX-2. And further, treatment with the STAT3 inhibitor could block such binding. So we proved that STAT3 could directly regulate COX-2 expression by binding in its promoter region. So we now prove that fetal-derived IGF-2 ligand could elicit implantation through STAT3 pathway to induce COX-2 expression. Finally, it's the working model of this paper. Wish you all could learn something from this presentation. Thank you all for your attention. That's all.